Welcome to Ask the Expert. Today we have the distinct pleasure of talking with Mia Øgaard Mønsted, and forgive me if I've pronounced that. She's going to be talking about phosphatidylcholine, a role in type 1 diabetes. Very interesting concept, one that I've spoken with a couple of people at the Bartholin Institute, particularly Karsten Bouchard, in the past, and it's really exciting to talk to her about her work. She's a trainee, or actually a newly minted PhD, and so she'll have some very cutting edge work to share with us. And I wanted to also have her just do a quick introduction about, give us a little background about herself. So welcome, Mia.
Thank you. Yeah, we have these Danish letters, yeah. So my name is Mia Øgaard Mønsted, and I got my PhD in November. I have been in Martin's laboratory at the Bartholin Institute for almost 4 years, where I've investigated the role of the intestine in the development of type 1 diabetes, where I've been primarily focused on intestinal permeability and these lipids that we're going to hear about today, and also a bit about gluten and type 1 diabetes. And I have a background in molecular medicine. And
Great.
where I am now, I start next month in a medical device company as a engineer.
Oh, fantastic. Very interesting.
Yeah.
And then I'm going to have, sort of just if Martin, can you illustrate a little bit about sort of the current focus of the laboratory?
Yes, of course. My name is Martin-Jorgensen, and I'm a group leader and senior researcher at the Bartholin Institute. Of course, our focus is type 1 diabetes, but we focus mostly on two promising areas in type 1 diabetes, namely intestinal permeability, which you probably know is increased in type 1 diabetes, but also in pre-type 1 diabetes and in family members to those who develop type 1 diabetes. So it seems to be a strong early factor in type 1 diabetes. So we look at that, which Mia will tell you more about. And then we also focus on virus, especially enterovirus, Coxsackie virus, and the innate immune system. We see that some important innate antiviral immune pathways are dysregulated in type 1 diabetes. So those are the two focus areas in the laboratory at the moment.
Fantastic. Yeah, these are very important and timely areas of research. So thanks for sort of sketching that out for us. Okay, so let's have Mia, take it away here and discuss her work.
First of all, thank you very much for inviting me. It's an interesting opportunity. I will tell you about one of my projects in my PhD called Phosphatidylcholine, a role in type 1 diabetes, where we'll present a paper that is currently that's currently submitted, and also some work from some of our students. So I will just briefly go over type 1 diabetes. I'm sure all you know what it is, but it's an autoimmune disease where you get a destruction of the pancreatic beta cells. So they stop producing insulin, and therefore the glucose levels in the blood is starting to rise, and you're in need of exogenous insulin injections every day, several times a day. Unfortunately, the incidence of type 1 diabetes is rising and has been for the past few decades. These are slides from several European countries. I'm pretty sure it's the same in America, where we see a steady incline of the incidence. There is a difference in incidence when you compare which country you are in. Finland has the highest incidence per 100,000 people of 51 persons, and I think it's China that has the smallest one, 0 to 1 per 100,000 persons. So it also depends on where you live, what your risk of
It's curious is that one of the early hypotheses was, the absence of sunlight, right? Or the that could be a possible hypothesis. But you've got Finland and Sweden, and then Kuwait is a fast third. So that's very curious that Kuwait would be third, isn't it?
Yeah, sure. It's not really my specialty, but it is. There is at least a lot of Scandinavian countries on top of this list.
Right.
Yeah.
Well, let's talk. Let's just plant that seed as you start to talk about the phosphatidylcholine, and have people thinking about how that might fit.
Yeah. So we have a great treatment for type 1 diabetes with these insulin injections, but unfortunately the mortality is still increased for patients. Here you see the life expectancy in the different age groups where you are diagnosed, and I think it's from Sweden. If you get diagnosed before the age of 10, you'll actually lose about 18 years of life expectancy, though. So that's quite a high number. And this is very much related to the complications you can get when you have type 1 diabetes, like strokes, but also retinopathy, kidney disease, neuropathy, and things like that, that also severely affects your quality of life. So we really need to know if we somehow can prevent this disease, and that is what we have been trying to do at the Bartholin Institute and have been interested in for maybe a decade or more in Carsten's behavior. Yeah. But to prevent the disease, we need to know the exact etiopathogenesis, and that is not entirely known yet, but we know that it's a multifactorial disease, which is where both genetics and environmental factors are important. On the genetic sides, we know that if you have a father, mother, or a sibling that has type 1 diabetes, you are in increased risk of developing type 1 diabetes. Furthermore, we have these HLA genotypes, which are very common, and about 90% of Scandinavian children with type 1 diabetes has one or both. And in monozygotic twin studies, about 20 to 50% of the other twin also develops type 1 diabetes. So for sure, the genetics are very important. But on the environmental side, we still from the monozygotic twin studies see that it's not 100%. So the environment must also be important. Also, the incidence is rising, which can't be explained just by genetic factors. And from migration studies, we have seen that when you migrate from a low incidence country to a high incidence country, your risk of developing type 1 diabetes, especially in young children, gets increased, which also suggests that the environment is very important. So the question is, where are we in contact with the environment? And one place would be the intestine, because everything we eat and drink and medicine and whatnot goes through your digestive tract and passes the intestine. The intestinal barrier has a very, very high surface area due to these villi protrusions in the small intestine, and also the microvilli on top of all of the intestinal cells, or the enterocytes at least. And if you fold them out, they can fill a whole tennis court. So there's a lot of place for environmental factors to bother you here. It serves as a self, non-self barrier through this thin epithelial lining, where you have different cell types, primarily enterocytes, but also goblet cells, which are producing this mucus layer, and for example, Paneth cells that are producing antimicrobial peptides. So in 2018, the perfect storm hypothesis was proposed for the development of type 1 diabetes to be involved with increased intestinal permeability, an aberrant intestinal microbiota, and an altered intestinal immune reaction, and that these would facilitate autoimmunity. So as I told you before, I have also done intestinal permeability experiments, but we will focus on this phosphatidylcholine, especially in the mucus. So phosphatidylcholine is a lipid class where you have two fatty acids, which can be of varying length and with several double bindings. And this confers both the function of the species, but also the hydrophobicity. Then it has a glycerol backbone, a phosphate group, and a choline head group, which is hydrophilic. So why are we interested in phosphatidylcholine in type 1 diabetes? Well, there has been a lot of studies trying to figure out if there are any metabolomic patterns that would be relevant in pre-type 1 diabetes patients, and if we could somehow do some kind of biomarker on these lipids. And this is a study from 2020 where he compares, this compares 4 studies and in pre-type 1 diabetes patients that develops type 1 diabetes later on and their lipidome in the blood. And what we see here is the consensus effect. So if a given lipid species has been observed several times, decreased, then it gets more blue, and if it's increased, then it gets more red. And you can see here for the phosphatidylcholines or PCs that is generally blue. So generally we have decreased levels of different phosphatidylcholine species in the blood prior to type 1 diabetes development. So what does this have to do with the mucus layer? Well, actually, phosphatidylcholine is supposed to associated with these large glycoproteins that are within the mucus layer and conferring a hydrophobic property here. So if we lack PCs here, then we might have a less hydrophobic and a less strengthened intestinal barrier. And this has been observed in ulcerative colitis patients that have severely reduced phosphatidylcholine in the mucus layer. And if they are given PCs in a capsule form, they actually improve their clinical index. They have also found that PCs are supposedly transported from the blood and into the mucus layer. And I say supposedly it's because it's there's really not a lot of data on this. These mucus lipids at all. But this is what we have been inspired by to see, well, do we have decreased levels of PCs in the mucus layer of type 1 diabetes? Of course, patients, but I have worked with mice, these NOD mice that develop spontaneously diabetes. Yeah, so our aim.
Can I interrupt for 1 second? Can we go back to that one slide?
Yeah.
So when there's a lack of PC, then does that result in sort of the mucus being less, strongly applied to the enterocytes, or what happens there?
Yeah, so it's a great question because we don't know much about it, but we know that if you put PCs on the intestinal surface, then you improve the intestinal permeability. So somehow we believe at least that less diabetogenic antigens can enter into the body. That's kind of the idea.
So but the mucus is perhaps anchored there more securely or something along those lines.
Yeah, exactly. So it associates with these large glycoproteins. So it's kind of an extra surface that is hydrophobic and has an extra barrier function.
Interesting. Okay, thank you for clarifying.
Yeah. Yes. So what I did in the part that is currently submitted was that I examined the levels of these PCs in pre-diabetic NOD mice, which is this mouse type that spontaneously developed type 1 diabetes, and matched them with B6 mice to have a healthy control. And then we also examined the associated plasma metabolome, because the PCs are transported from the blood. And then I will also present some data on supplementation with PCs and if they can alleviate autoimmune diabetes in NOD mice, and some data on a macrophage cell line that is and how PCs can dampen the LPS response in these. Yes. So this is the experimental setup where we have the NOD mice and the B6 mice at three ages here before they develop type 1 diabetes. And here we took out different organs and so on. So I will start with the mucus layer. So I scraped off the mucus layer. That's also kind of inspired by these ulcerative colitis patients, but you get all of the lipids that are within the mucus layer, also from enterocytes and also from the MUC2 associated lipids. And then we analyze them with a method called shotgun lipidomics, which is very precise, where you can measure about 400 different lipid species, but we concentrated on these PCs. In the jejunum, we saw that on the class level, meaning the total sum of all of these different species, there was a decrease at 4 weeks of age. And it's not as decreased as in the ulcerative colitis mouse models, but it's still there and it could have an effect on the hydrophobicity. Then we examined if it was caused by specific species. So this is a full change plot where you only see the levels in the NOD mice. So the B6 mice are the black line. But as you see, there are not many that are statistically significant. So it suggests that it's more or less all of the species that are decreased here at 4 weeks of age, but not at 13 weeks of age. In the colonic mucus, we saw no alterations in the at the class level, but instead we saw actually on the species level a lot of different species that were decreased at both 4 and 13 weeks of age, as seen here on the full change plot is primarily decreased. So that could mean that there are some specific species that have functions that are currently unknown, but that they have an effect on the hydrophobicity or the signaling properties of the mucus layer. That's at least our hypothesis. Yeah. Yes, sorry, I just remember what the next was. We wanted to see if these changes were caused by changes in the thickness of the mucus layer. But as you see here, we saw no difference in the amount of goblet cells between the NOD and the B6 mice, or the thickness of the inner mucus layer in the colon, or the area. So the alterations were not caused by differences in the histology of the mucus layer. But we supplemented these results with metabolomics on plasma, because we know that the PCs are transported from the blood into the mucus. And here, with focus on the PCs, we saw decreased levels of 3 PC species at young age here, 34.1, 64.4, and 38.6, and 1 was increased in the NOD mice. But what I noticed was that this specific species called PC36.4 was also decreased in the colonic mucus at 4 and 13 weeks of age. And if we go back to this consensus plot, this is actually also the only PC species here, 36 length and 4 double bindings, that has a consensus effect of -3. That means that 3 studies have shown that it's decreased at the pre-diabetes. And furthermore, in a recent study, they have seen in patients that go from being islet autoantibody positive to islet autoantibody negative, that it actually has an increase of this specific lipid species, suggesting that it could be some kind of relevant biomarker in type 1 diabetes. patients also. So with these lipid species, we'd also got a lot of metabolomics results, and I won't go into very much detail. So this is the comparison between NOD and B6 mice. What we saw was an increased level of ketone bodies as a result of beta oxidation, increased levels of branch-changed amino acids, which are associated with beta cell dysfunction, and also these acyl carnitines, which is also a byproduct of beta oxidation, were also increased in the NOD mice. And this is kind of explainable by we are comparing a type 1 diabetes mouse model to one that is not. So we would expect there was some metabolomic results that were differing here. So we also did a Cecal microbiome analysis by sequencing the variable region 1 to 8 of the 16S rRNA in the Cecal content using the Minion. And we did that because we know that the composition of the microbiota is important for the mucus layer. And it's also been a lot of studies in type 1 diabetes patients where they see altering microbiota. So here we see that we have an altered beta diversity. So we have a significant difference between the microbial communities in the NOD mice here in blue and the B6 mice here in red. So, and it also alters with age, which is fairly normal. When we look at the species that are driving this difference between the mice, we can see here a lot of species. I will only go into a little bit of detail here. This specific species, Aneotronchus, was completely absent in the B6 mice, and Alobaculum and Tuisobacter was completely absent in the NOD mice. And these three species are both negatively and positively associated with short-chain fatty acids. And short-chain fatty acids are beneficial for the gut, and it also induces a tolerogenic immune response, and short-chain fatty acids are also decreased in type 1 diabetes patients. So we see kind of the similar here, at least from this perspective. So this was what we have currently submitted, and hopefully we will get it out soon. So I will jump to the next experiment, where we wanted to see if we could supplement the NOD mice with PCs. So we got a PC cocktail, which is derived from eggs, and also choline because it's a precursor to PCs, and then supplemented into the diet. So we gave this diet from 3 to 4 weeks and until the end of the experiment, about 217 days. And this was, of course, because we have these observance of decreased levels of PCs. So it could be a relatively easy experiment to see what effect does this have on the diabetes incidence. But unfortunately, we were not able to see any difference in diabetes incidence. As seen here on the incidence graph, they developed type 1 diabetes at the same time and got more or less the same incidence. And this could be, of course, just because that's how it is, or because we are not supplying in the right way. Maybe it should be directly into the veins, or because it's a cocktail of PCs. So we know from other experiments that these specific species, like 36,4, have specific functions. So maybe this cocktail doesn't have the correct distribution of lipids. So one of our master students conducted a cell experiment concurrently with the mice experiment just showed, where he stimulated macrophages with PCs, either the cocktail that was also incorporated into the diet, or 36,4 because of our interest in that. And then he removed the PCs and stimulated with LPS, conducting an inflammatory response. So what he saw was that in the in the medium, he measured NO, which is an inflammatory response marker. And what he saw was that when he doesn't treat with LPS, we don't get any NO. If we treat with a physiological level of LPS, then actually 36,4 is able to reduce the NO response, also to a higher degree than the cocktail. And if we are in very high levels of LPS, then both the cocktail and the 36,4 reduces the NO response. And he was able to verify this with qPCR on INOS and also TNF-alpha, which I have on this slide, where you can see also without LPS, the 36,4 is actually reducing INOS levels in the cells. And also here with the treatment with LPS. And interestingly, he also observed that when he treated with 36,4, he actually got an anti-inflammatory response with the production of IL-10 without any LPS. So it was not strong enough when the LPS stimulants were also on it, but he got a pretty strong response when they were untreated. So 36,4 definitely has anti-inflammatory properties, which has also been shown by others, which is kind of interesting when you also think about the current literature of type 1 diabetes and this specific lipid species. So to conclude, in the large mouse experiment, we saw these aberrant or decreased levels of PCs in the mucus layer. And they were kind of related to plasma levels of PCs, or not to the same degree, but at least we were able to focus on this 36,4, which is relevant for human type 1 diabetes. It was not related to alterations in mucus histology, but we also observed alterations in the microbiome, which were related to short-chain fatty acids production, which is also seen in type 1 diabetes. Unfortunately, the diet supplementation with a cocktail of PCs did not reduce diabetes incidence. It could, of course, be very interesting to see if the 36,4 would have the effect, but it's very expensive to have such a clean, it's not called clean, like 100% PC 36,4 is very expensive. Yeah, and then we know that PC 36,4 has anti-inflammatory effects in this macrophage cell line. So we, of course, think that there should be more research on PC 36,4 in type 1 diabetes. Maybe it can be used as a biomarker, or maybe it has therapeutic potentials, either by ingesting it or through IV or something like that. And then we are currently, some of our other students are currently seeing if it has an effect on other cell lines, in particular beta cells and their insulin production. So thank you for inviting me, and I of course want to thank my team at the Bartholin Institute and my collaboration partners and my funding bodies.
That's fantastic. Thank you very much, Mia. I wondered if anyone had thought about doing the experiment where you replace or somehow enhance the missing bacterial species and then add the PC. I wonder if that might, what do you think about that?
Yeah, that could be interesting. I'm really, when you, these ulcerative colitis patients, they get a specific capsule that is only degraded in colon, and I would really want to be able to do that also in mice. I think that's not really so easy because we just supplemented it with the diet. So it might have been broken down in the small intestine and not reach the colon. I know there has been a lot of study manipulating the microbiome, specifically inducing short-chain fatty acid production through fiber diets, and it has a positive effect on the diabetes incidence in not mice at least. So that could be interesting, yeah, to combine it, yeah.
Yeah, it's fascinating. I think that this area has been kind of like understudied, but this paper is really interesting that you've got here, and I think it's going to pique some interest and really kind of get people thinking about approaching this whole paradigm, more robustly. I didn't know if you, Martin, want to join in at any and have a few and have some input in terms of like what's going to happen. You're heading out to your next stage of your career. I mean, is there, what's happening after you go in the laboratory, or?
Yeah, I just wanted to add that it's just strange because many groups has actually been looking at dyslipidemia and type 1 diabetes and see reductions of many of the same species as MIA see reductions in, but not very many are studying the specific PC species. But they seemingly have a strong anti-inflammatory effect. So I think it's important to try and characterize these PC species that we see reduced both in serum and in the mucus layer in pre-type 1 diabetes. I think it's important to focus on this pre-diabetic phase, right? Because the things we see changed in that phase. Yeah, there is a bigger potential to prevent type 1 diabetes with these things.
Yeah, absolutely. And with the advent of teplizumab and its acceptance here in the US by the FDA, right, that also, kind of incentivizes people to look very closely at the prodrome and what's happening as people head towards the diagnosis of diabetes, so that perhaps there might be, multi-drug approaches to stop that progression. I wanted to, I also found that it was fascinating that there was no histological changes when you looked at the NOD mouse in the gut. I wondered if you know if anyone has done the work in human samples that are associated with type 1.
Lipidomics in the mucus, or?
Well, looking at the sort of you guys looked at the histology of the gut, and you noticed, there was no real change in the number of goblet cells or the thickness of the mucus. And I don't know, I haven't seen that. I just didn't know if you did, because it's more in your realm of research.
I don't think there is any mucus histology out there from type 1 diabetes patients. No.
Yeah, it would have to be the samples, right, of the post-mortem samples.
Yeah, in general, but it's also the mucus layer is very difficult to perform histology on, because it's just gets ripped off in the colon. I measured the inner mucus layer, which is also very commonly done, but the outer mucus layer is also important, but you can't really analyze it like that. Yeah.
Were you going to add something, Martin?
Yeah. Well, in general, we do see changes in the intestinal in the intestine in NOD mice, but not that much in humans. We see enteropathy in the pre-diabetic phase of NOD mice, which we can reduce, for instance, with a gluten-free diet. So we can actually increase the surface area. We can increase the villus to crypt ratio in the small intestine with just avoiding gluten in utero. So, but I don't think that the same changes are present in human type 1 diabetes as we see in the NOD mice.
Yeah, I just didn't know about that literature, whether or not people had extended the work into human samples. Well, there's room for inquiry, I guess. So this was really great, very interesting. Thank you very much for presenting this work, Mia. It was really, really fascinating, and I think that it'll be viewed and talked about, I think, in some circles that are really looking at this interface, which is an important one.
Yeah.
Thank you very much, and I hope you guys have a great rest of your day, evening.
Thank you.
Yeah.
Bye-bye.
Thank you very much.
Yeah, thank you. Bye-bye.
Bye-bye.
Bye.